decisions. It wasn't coronavirus.
Flu is going around though. Dr. Worrell was sick, his kids were sick. You guys all been vaccinated for flu? Good.
Not mine. Easy. Just wrote down everything I knew.
What was this?
The biochem quiz over Dr. Walters.
Great. There it is. All right. Is everybody here? Who are we missing? Oh, yeah. I'm so close to that prairie dog that you can see my reflection in his eye if you look closely.
Is your pictures that are your screenshots?
Yes, that's Komodo dragon from the island of Komodo.
Yeah,
In 2010, I was there for the Association for Tropical Biology and Conservation meetings, and I went out a week early to travel around Komodo and Flores, which is where Komodo dragons live, and then I stayed for a week after that and went to Java and Krakatoa, which is this island off the coast of Java that blew up in 1883 in the largest volcanic eruption known to ever occur. Went on the island that was formed in its aftermath. It's actually formed and reformed a number of times, and it's actively erupting while I was there because it's been very active for a long time. Hiked around a section of Java. Yeah, so all the pictures that appear on my screen are pictures I've taken. This is a spiny soft-shell turtle from Florida. Anyway, that is not what I want to talk to you about. I want to tell you that I have your lab reports graded. I'll give them back to you at the end of class. I also want to talk to you about the next lab report coming up. So as I've talked to people who have started working on their lab reports that are due right after spring break, the Tuesday after spring break, I think some of you are struggling with how to talk about the results of the whole thing. And so what I want to do is just take a couple of minutes to kind of get across what I'm thinking of. So when we think about how a predator gains prey while it's out foraging, you can think of it as the number of discs captured as density goes up. This is going to largely be affected by three things: handling time, detection probability. And the third thing is the spatial distribution. So normally we would think of this curve as reaching an asymptote. What imposes that asymptote on the number of prey? Why can't you, if there are 256 prey in your environment, why can't you just catch all 256 prey? Okay, so there's inherent in the shape of this curve a handling time constraint. As handling time increases, what happens to the curve?
It increases at a progressively slower rate.
Okay, so as density increases, number of disks increases, but if handling time is higher, it increases at a slower rate, meaning that the overall elevation is lower here due to handling time constraints being higher, i.e. handling time is longer, handling time shorter. If we reduce handling time, what happens to this curve?
The elevation increases.
Elevation increases. Does much happen to it down here at this end? Not really, because there's so few prey items there that you spend all of your time in search, you spend relatively little time in handling, and so things kind of stay the same down here, but your elevation that you reach is ultimately higher until eventually you just don't have any handling time constraints at all, and then you're catching them directly in proportion to their density. So as you're writing your introduction, write it in a way that you kind of get across the idea of how does handling time affect the number of prey that a predator is going to get in a given unit of time. When we think about detection probability, we still have this shaped curve. Why?
Because the amount of time it takes to detect the prey constrains the.
No. I mean, yes, it does, but why is it this shape? Just because the prey are harder to detect, what do you still have to do once you catch prey? You still have to handle it. So the shape of this curve is still affected by handling time constraints. Because just because something is easier to detect doesn't mean that it's any easier or harder to handle once you've detected it. Now, what else can then affect the elevation of this curve? In terms of detectability, if prey are harder to detect, what happens? Yeah, lower elevation. You just get fewer prey overall because in addition to handling time constraining you, you're also now constrained by the fact that it's hard to detect prey. And if prey are easy to detect, then this curve is higher. So this is low probability of detection, high probability of detection, and medium, if you want to do it this way. When you clump prey, rather than having prey that are uniformly distributed, what effect does this have on things?
It again lowers the elevation.
Once again, lower elevation under clumping, why?
Because of the time it takes to move from clump to clump.
So when you hit a clump, you forge around in there, and then once you leave that clump, once you think you have depleted that clump, you then have to spend time searching. Because they're clumped, there's more space to search because the prey are clumped. Is that issue going to be more of an issue at low densities or high densities? It's It's gonna be more of an issue at low densities because those clumps can be further and further apart because there just aren't that many prey in the environment. So you might expect this to have more of an effect at low densities than at high densities, but the general, once again, the general rule should be that you should see, once again, lower total number of prey captured when they are clumped than when they are uniform because of travel time between patches, essentially. Yeah, Alexis.
Since in all of the experiments, the prey were uniform except for the clumped one,
Yes.
which one do you think would be most valuable to make the comparison to?
Finger sandpaper search, because you were searching with your finger, searching for sandpaper discs. So for spatial distribution, it would be finger sandpaper, because the only difference between finger sandpaper and the type 3, what I have labeled in your R script as type 3, is this is uniform versus clumped.
Okay.
So that would be the most appropriate thing to compare it to. They both have handling time constraints because everybody was still putting their discs in the bag for both of those. So the only difference here really is uniform versus clumped. So when you write your introduction, in particular, at the end, you can say, well, if we do things that will increase or decrease handling time, these are the effects that we should expect to see. If we do things that influence the detectability of prey, it should have these effects. If we do those two things in concert, then they should both have more an effect than doing either of them separately. Yeah.
On the specifications, you said that you wanted us to relate the hypotheses to the functional responses that they would produce.
Yes.
Is that still kind of what?
Yeah, that's exactly what I'm drawing you towards.
So we would say something like, oh, if we introduce a handling time constraint, it'll produce a type 2 functional response.
Yeah, and all of these basically look like type 2 functional responses. We didn't really produce what is credibly a type 1 functional response, nor did we produce credibly a type 3 functional response. They all ended up
But we
looking like type 2.
hypothesized that we might.
Yes.
Okay.
Yes, we did. And then we didn't observe those. And I've talked to you in my office about the reasons why we might not see the results that we were expecting. So I'm just trying to give you some pointers on kind of how to organize the way you talk about these things. Because they all kind of show the influence of handling time, but to lesser degrees, depending on what the specifics of any given set of trials were. We didn't really observe a type 1 functional response, which in theory has no handling time associated with it at all. All right, any questions about that as you get ready for that? This next part, I wonder whether I should tell you about this or not, but you are all juniors and seniors, and so you should be adult enough to handle this. Since we're kind of waiting on migration to kick in, I was just looking at some migration maps the other day and birds that would normally be up here. The kinds of birds that we're looking for up here are still down in Texas and southern Oklahoma and southern Arkansas. Since we're waiting on migration before we can put the snake models out, we have a little bit of breathing room in here. So what I want to do is, your paper is due Tuesday after spring break. I still want to make that due Tuesday after spring break, but what I want to do that Tuesday is I want you guys to trade papers with one another, do peer reviews, just one paper each, review one of your peers' papers, and trade with whoever you want to, and then have it back, that's Tuesday, have it back to the person by Thursday so that they can make revisions so that a final version will be due Tuesday the next week, which I think is the 31st of March. We have plenty of time for the rest of the things that we're doing in lab. This will allow you to give you some feedback from somebody other than me, and also might give you other ideas of how to go about writing things. When I tell you, when I give you advice on writing, sometimes it's not terribly helpful because I say, okay, I'm not really caring for this writing. This writing could be tighter, it could be better organized, et cetera, et cetera. Sometimes seeing the way other people write is useful to you. Not that you're supposed to copy off of other people's ideas or copy what they're writing, but the more writing you read, the better off you are. So there was a guy who came and gave a talk at William Jewell College a number of years ago, and in his oral presentation, to the students, he basically did this thing where at the end of his introduction, he told us what his hypotheses were, and then he showed us a made-up graph of what the data should look like if the hypotheses were actually true. And then when he went through and presented his results, before he showed us the actual data, he flashed that figure back up and said, just to remind you, this is what the data should look like if the hypothesis is correct. Now, here's what the data actually look like. And I thought that was a really useful thing to do, both at the end of the introduction and in the results section. And so I've incorporated doing that into my own way of doing things as I make presentations. I've also incorporated that into grant proposals and things like this because it makes it clear to a granting agency that I do have a clear idea of what things should turn out to be like. So I stole a person's idea, although I didn't steal their exact form of that because he was a plant ecologist and I'm an animal ecologist. So you get ideas for different ways of doing things as you see more people doing things. So if you guys are okay with doing this, I would like to do this. Do you think this would be useful to you? Okay, what I struggled with prior to coming in and telling you this today was if I tell you that it's really not finally due until this date, you might show up on this date with a bunch of crap in your hands, which doesn't serve anybody any good. It doesn't serve you any good because you get a review on what's a not very good paper, and it doesn't serve the other person any good because they don't get to see what your best work is. So what I would like you to do is come back from spring break with your best version, the version that you would have turned into me. Now, when you get your grades back today, you're gonna see a number of you passed, but then there are also comments on there on ways in which you could be improving your writing. This is an opportunity to get some feedback from somebody other than me on, for example, how to improve your writing. We did this to you in evolution ecology with your final paper. Do you recall what we did to you back then? No? You forgotten about it? What?
No, we forgot.
You forgot. We made a due date. You all showed up with your papers, and then we sat you down in the lab and we did, well, Luke should remember, this was just last semester. Yeah, you showed up on the day that your paper was due. We sat you down in the lab and you did peer review, and then your final version was due the next week. The reason we do that with first year students is because, quite frankly, we don't trust them. If we tell them that it's not really due that week, they will show up without a fully formed paper. And so we trick them into saying it is due this week. We then do peer review, and then they get a week to revise it based on the reviews that they get back from their fellows. We also collect a copy of it ourselves that we turn around in just a couple of days so that they also have some preliminary feedback from us. I'm glad that you purged that from your memory because we're always worried that the upper class students will tell the first year students and then they won't show up with it with a full thing. So if we're gonna do this and for this to be effective, you need to be ready to turn in a really good version of your paper on that Tuesday, otherwise you're just wasting everybody's time and I would rather that we not waste one another's time. And then you get some feedback quickly so that you then have like 4 days to crank out whatever changes the person that you're reading, who reviewed it for you, has suggested to you. You also have some time to incorporate other ideas into the way that you're thinking. You'll have a copy of the rubric so you can all also pay attention to whether or not somebody has missed something fundamentally important that is in the rubric. So, sound like a plan? Right. If you'll find it useful, we'll do it. If it ends up not being useful, then we'll scrap it. We'll just be due. I can still make it due the 31st, give you a little extra time. There just won't be any peer review.
I'd like to do peer review.
I do too.
Likewise.
Okay. Then we'll do that. All right. questions from the last time?
You told me to remind you to explain the Concord fallacy,
Oh yeah, why did the Concord fallacy come up?
because I said, why don't the birds just like not feed the cuckoos and leave?
Yes, so there's a really good reason for why host birds don't feed the cuckoos, in spite of the fact that sometimes the baby cuckoo may be 3 times bigger than the host adult bird. And the reason for this is that organisms have a tremendous drive to reproduce. Part of that drive to reproduce is if you have an offspring sitting in your nest, the rule is basically you should feed it. And oftentimes this is going to be hardwired. There's a whole slew of experiments that have looked at sign stimuli. We haven't really talked about sign stimuli. Who can tell me what a sign stimulus is? Anybody? Yeah.
I mean, I can maybe think of an example, I don't know if it's accurate, but you just like display a certain symbol or image or something that causes somebody to remember something or to.
That's true. And that one just didn't work. That's a shame. Maybe I faked it too badly.
You want us to yawn too?
I did want you to yawn also. So a sign stimulus is this kind of stereotyped response to a particular stimulus. So a guy named Konrad Lorenz, who was one of the founders of the field of animal behavior as we currently think about it, he painted sticks white, and then on the end of the stick he put a little red dot, and he used this to basically feed a species of gull that has the parents that have a beak that is white with a little red dot on the tip of it, and he poked this at little baby gulls in a nest, and when the baby gulls saw the white dowel with a red dot on it, they opened their mouths and started begging for food. And these were baby gulls, so they really don't know much of anything. And so this was a sign stimulus because whenever a baby sees the beak of their parent, that's a sign that they're about to be fed. And so what you should do is you should open your mouth and prepare to be fed. And so sign stimuli are these kinds of stimuli that evoke a response that is really kind of outside of the control of the individual evoking the response, producing the response in response to some sort of stimulus. So oftentimes a begging offspring serves as a sign stimulus to a parent, such that when you see a baby bird begging, your response is to feed it whatever food you have ingested yourself. And what birds typically do is they regurgitate their food into the mouths of their babies. And they will do this regardless of how much the baby bird doesn't look like you in any way. So that's one reason. Another reason might be what's referred to as the Concord fallacy. Do you guys remember the Concorde airplane? It went out of commission, I think, probably before most of you were born. The Concorde? Nobody? Nobody?
The one that went to like New York and London in like 3 hours.
It went from New York to London very fast because it was a supersonic. It was a supersonic.
Yeah, it was like too powerful for the engine was too powerful for the plane. Yeah, like ripped it up.
Supersonic passenger jet. It looked like this. It had a pointy nose. It had gull wings. It had gigantic engines. People in New York complained about it a lot because there would be these sonic booms that went off over New York all the time when the plane was flying in and flying out. It was developed, I forget what company developed it, but it was a French company, and the French company that developed this was subsidized by the government. So the French government put a lot of money into the Concorde, and one of the things that happened with the Concorde was they fell behind in terms of production goals, and they also experienced lots of cost overruns. And every time they would have a cost overrun or a delay in production, they would go back to the French government and they would say, we need more money because it's costing more to develop this thing than we thought. And the French government gave them more money, and then they used that money, and then they ran behind, and then they went back to the French government for more money. And the French government said to themselves, well, we've already put $1 billion into this, so if we get out of this now, we'll lose all of that investment. So they put more money in, and now they're like, well, if they need more money, we should give it to them, because now we have $1.5 billion into it, and if we quit now, we'll lose that $1.5 billion of investment. And then it runs behind more, and there are more cost overruns, and now they're asking for another $500 million. So now they're like, well, if you don't give them another $500 million, we will lose, the $2 billion or whatever that we've already put into it. And they did this over and over and over again.
Into developing the plan?
Into developing the plane, yes. And the plane finally got built, but it never really gained back all of that investment. And so the Concord fallacy is this idea that you should invest in something because you have previously invested in it. And oftentimes animals make non-optimal decisions in terms of their behavior because they commit the Concord fallacy. This idea that I've laid an egg, I'm a female host bird, I've already invested a certain amount of my body mass in producing that egg, I had better incubate it because I don't want to lose my investment. And then it hatches earlier than you thought it was, and the chick is way bigger, and you're like, that chick doesn't look like me, but I've already now invested all of this energy in producing the egg, and also all the energy in sitting on the egg, reducing my own feeding rate so that I can get that egg to hatch. So I need to keep investing in this chick, despite the fact that it looks really weird, because I already have all of this investment tied up into it. And then over time, you are just more and more invested in it, and so you tend to put more and more future investment into it, even though it doesn't serve you any reason. And so we see in the area of life history theory, oftentimes individuals make investment decisions that are suboptimal because of prior investment, protecting prior investment. This happens with people in the stock market. You've invested in a stock in the stock market, and people make all sorts of irrational decisions in terms of how they invest in the stock market based on kind of their prior investment strategies. In some cases, it's really good to turn your back on an investment so that you don't lose more money than you already have lost money. And in some cases, it's prudent to just stick it out. So for example, if you are heavily invested in coal stocks, I would just get out of coal stocks. Coal is never going to recover. Natural gas is way too cheap. Coal is way too dirty. Like, in spite of any president's claims to the contrary, the coal industry is not ever coming back. So if you are invested in coal, you should just say goodbye to that and lose whatever money you've already lost on coal rather than holding on to it thinking that someday coal is going to turn around. There aren't a lot of stock options that I can look to in the 21st century here and say that firmly about, but I feel pretty safe about coal on this front. So people commit the Concord fallacy, governments commit the Concord fallacy. Animals raising offspring are especially prone to committing the Concord fallacy. And it's the mistake of continuing to invest when what you should do is kiss that investment goodbye and invest in a new effort. If you're a host bird with a brood parasite in your nest, it's so much better to just let that bird die, go build a new nest someplace else, and lay some more eggs. But we see that they don't do that because they're prone to, they know what investment they've put into that reproductive effort, and they don't have the cognitive abilities to think their way out of that. The Concord fallacy. Does everybody understand what the Concord fallacy is? All right. Okay. It's not in the textbook anywhere, by the way. I had trouble interpreting this graph, so in the time since we last met, since Wednesday, I went back and read the paper, the original paper that this came from, And so this is the cuckoo thing, the cuckoo study where they basically quantified the color of the eggs. And they quantified two things. They quantified the background of the eggs, and then they also quantified the spottiness of the eggs. And I told you that I wasn't understanding this figure. And the reason was I wasn't understanding it is because of relying on nothing but the figure to figure it out. And then I read the paper. What they're basically showing is a species' tendency to reject eggs versus the percentage of overlap in the ground color of the eggs that is shared between host and brood parasite. And what it's showing is kind of the coevolutionary nature of this. If you're a brood parasite and you live with a bird that doesn't tend to reject your eggs, there's no selection for you to be a precise mimic of those eggs. And so we see that the dunnock has the least amount of overlap, and that's presumably driven by the fact that dunnocks don't ever reject eggs. So if you're laying your brood parasitic eggs in the nest of a bird that doesn't reject eggs from its nest, there's no selective advantage to have precise mimicry. If you are a brood parasite and you are laying your eggs in a species that tends to reject eggs a lot, then there's selection acting on the brood parasite to produce eggs that are precise mimics of the host bird's eggs. And this only shows you a certain number of the species that they looked at. But they actually looked at a lot of different species. So here's the figure from the paper. And so they looked at a number, and you can't see. They actually looked at more species of eggs than are in the figure in the book, which is why there are more species on that scatter plot. And so, as it turns out, there's more to the scatter plot. This is the scatter plot that is in your book, showing that in species where there's a low rejection rate, there's really imprecise mimics or lack of mimicry altogether. And in species where they reject eggs a lot, there's a higher level of precise mimicry. But this is mainly due to the ground color, the background color on the eggs. When you look at the coloration of the spots don't really matter at all in terms of how much spotting, what the coloration of the spotting is, or anything else. And so it really has to do mainly with the background color of the eggs more than anything else as to what provides a signal of whether or not you're doing precise or imprecise mimicry in this case. So, that clarified that. Does everybody understand that figure better now? Okay. Last thing I want to talk to you about is another situation that is presumably a predator-prey arms race. This involves an interaction between bats and noctuid moths. This is not in your textbook. So noctuid moths, Noctuidae is a family of moths, the underwing moths here. On either side of the body, there's a tympanum. And the tympanum is basically a thinning of the exoskeleton. And underneath this tympanum, there are these air sacs. And as sound strikes the tympanum, it vibrates the air sacs underneath. And these air sacs, as they vibrate, stimulate two sensory neurons. So there's an A1 sensor and an A2 sensor. These sensors go off to the thoracic ganglia, and then they interact with interneurons, and then the interneurons go to the brain, and that information gets processed. The A1 and A2 receptors respond differently to sound. Yes?
Okay,
Yeah? What's the question?
I got exoskeleton thinning with the air sacs underneath, and then I lost you.
So this is on the outside. So this is the outside of the moth. This is the inside of the moth. They're located on the thorax. They're right here and here. Underneath the tympanum are air sacs. As the tympanum is vibrated by noise, it vibrates the air sacs. As the air sacs vibrate, these two nerves get activated. They depolarize due to the mechanics mechanical stimulation of the air sacs vibrating. Very similar to what your ear does. The two receptors respond differently to different intensities of sound. Or they respond differently to different intensities of sound. So the A1 receptor, this is the A1 receptor if I'm recalling correctly. The A1 receptor responds to low intensity sound at lower frequencies than it responds to high intensity sounds. And so if there's a really high pitched sound, it only responds to that high pitched sound if that sound is also loud. If it's low frequency sound, they only detect low frequency sounds when they're not very intense. So the y-axis here is intensity of, the y-axis here is intensity of sound, and then this is the frequencies of sound that they respond to. And
So you said it responds to low intensity at low frequency?
low intensity sound gets responded to in terms of low frequency. So peak responses for these things are at low intensities, and high intensities, I'm not sure that I'm saying this correctly.
Higher frequency, higher intensity.
Yeah, so the kind of sound that you respond to, so if it's a really soft sound, it's only going to respond if the sound is low. So the tympanum vibrates more in this range of sound, low frequency sound, if that sound is at low intensities. It only reacts to high intensity sound if that sound is intense, or sorry, high frequency sound if that sound is also intense. So
low pitch and soft,
Low-
high pitch and loud.
low pitch and soft, high pitch and loud. Why high pitch and loud and low pitch and soft? Low pitched noises are going to be just general noises of the environment for the most part. And oftentimes, because you're a flying insect, those noises are not going to be particularly close to you. High pitched sound, though, is something that you need to pay attention to. And high-pitched sound is especially important if that high-pitched sound is intense, because these things get fed upon by bats. And bats echolocate. They find their food using echolocation. And so they send out really high-frequency sound, really high-pitched sound, and it's directed forward in a way that also makes it really intense. Now, you don't hear this because you can't hear in the frequencies that bats emit, but moths can hear in the frequencies that bats emit. If you go out to a place where bats are flying around and you record sound on a recorder that can record high frequencies, and then you play that sound back slowly, you can start to hear the chirps of the bats, because slowing the tape down generally lowers the frequency of it so that you can now hear it audibly. So echolocation by bats is really high pitched and generally really loud, directed out in front of them, because they're waiting for the sound to bounce off of objects to basically tell them where objects are. Experiment done in a room kind of like this, when they first discovered echolocation, is they put a bat in a room and they strung piano wire all over the room, crisscrossing, up and down, across, in all kinds of crazy patterns, and then they set the bat loose in the room. And what they discovered with the bat is that in a room with crisscrossing piano wires, the bat could fly around and didn't strike any of the wires. It's chirping the whole time. The scientists can't hear the chirping, but they know that they emit these ultrasound chirps. Then they put the bat in the room, and then they played over speakers ultrasound, And as soon as they did that, the bat started flying into the piano wires, got knocked to the ground, and actually didn't try to get back up because it couldn't navigate effectively, thus proving definitively that bats do use these ultra high frequencies for echolocation purposes. It was a cool experiment that you probably couldn't get away with anymore, but it demonstrated the concept really well. So what happens in the moth is as a bat is flying around, it's sending out these exploratory chirps. Chirp, 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 chirp. When these noises hit the A1 receptor, the A1 receptor responds to low intensity stimuli. The A2 receptor is less of a responder to low intensity stimuli. Middle, moderate intensity stimuli, once again activate the A1 receptor. The A2 receptor doesn't respond at all. The A2 receptor doesn't respond until the intensity of the stimulus gets high enough that it gets activated. And so the way in which the tympanum responds to low intensity and high intensity sound has nothing to do with the structure of the tympanum or the air sacs. It has to do with what are the stimuli that influence and activate the A1 and the A2 receptor. A1 responds to all intensities of sound, really loud sound as well as really soft sound, and the A2 receptor only responds to really loud noises.
Are these red lines?
Yes. Those are depolarizations essentially.
So like all of them together show more of a.
Yeah, so low intensity sound, A1 is activated somewhat, and then after the sound goes away, it goes back to kind of background levels of activation. A2 doesn't change. A1, high levels of activation during sound emission, then it goes back to background. A2, no change. High intensity stimulus activates A1 at higher levels than for moderate intensity, but A2 goes from not being activated at all to being activated a lot, and that activation is just how many nerve impulses is that receptor sending per unit time. Yeah.
So you said A1 here is activated under all intensities, and
Yes.
A2 is only under really loud
Yeah.
intensities, but I thought you just said on the previous slide that A1 responds to low intensities.
This is actually the entire tympanum.
Okay, so it's A1 and A2.
A1 and A2. And what makes the differences in low intensity versus high intensity is which of these two receptors is actually being stimulated. What a bat looks like to a moth is burst of sound, burst of sound, burst of sound, burst of sound. If you play that same frequency and intensity of sound at the beginning, the neuron will react to it, but over time, the neuron basically stops reacting to it. Presumably what's happening is like any nerve, if you overstimulate it, it loses its ability to continue generating depolarizations because it takes time to repolarize the membrane after it gets stimulated, and you eventually just exhaust its ability because of the number of sodium potassium pumps that it has to restore that polarization on the neuron, so that it just goes back to a slow rate of depolarization because you've essentially overstimulated it. You guys study this in physiology.
So the top, the ultrasound pulses is what the bat is doing.
Yes.
And the body.
This is
what the
sound
bat's not
from the
doing.
bat. This is a response of, in this case, the A1 receptor in the moth.
Does the bat do the bottom thing?
No, the bat doesn't do the bottom thing, generally speaking. This is some researcher just basically playing steady ultrasound to see if you give it a continuous ultrasound, is it going to continue to be activated at the same rate that it was being activated before? And it doesn't. It gets overstimulated. Yeah.
Why didn't they perform the same bottom experiment on the A2 receptor?
They did, and they both respond the same way.
Oh, okay.
So, because you have tympana on both sides, you have A1 and A2 receptors on both sides of the moth can actually locate where in space the bat is, depending on how the two sides of the body are receiving sound. If the sound is off to the side, what you will see is that the tympanum on one side responds more intensely than the tympanum on the other side, which is responding less intensely because the bat is on one side of the moth than on the other side of the moth. If the bat is above them, the tympana are exposed more when the wings are up as it's flying. As a result, it gets more stimulus, and then when the wings are flapped down, the wings are covering the tympana, and so it knows that the bat is above it rather than below it. And if the bat is behind it, you'll get even stimulus on both tympana, basically indicate that the bat is behind it. What generally happens when a moth first hears a bat is it uses the varying inputs to the two sides, the two tympana, and it orients its body in such a way that the bat is now behind it, and it just flies in the opposite direction. Fly away, fly away. As the bat gets closer and closer, the bat is putting out pulses. The bat is putting out pulses. Chirp, 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 chirp. As the moth hears this, the moth turns and flees, flies the other direction, but oftentimes the bat flies faster than a moth. Chirp, 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 chirp. You start noticing here that the frequency between chirps begins to decrease because the bat is beginning to get echoes reflecting off of the moth. When it knows that it has a moth in its sights, so to speak, it picks up the frequency of chirps, and it also increases their amplitude, how loud they are and how close together they are. The faster together they are, the more sound you're putting out there, and you're getting more feedback back, and the louder it gets, once again, the better you are to see with your echolocation what's out in front of you. So they alter their vocalization behavior so that during this part, when they've detected a moth during this part, they can get a very fine scale position on the moth because the moth is continuing to move throughout all of this also. So if you continue this rate of sound emission, the moth can move a lot, can change position a lot between this vocalization and this vocalization. The moth moves very little between this vocalization and this vocalization, moves even less between this vocalization and that vocalization. Alexis.
So are you saying it chirps more when it gets closer?
Chirps more frequently and louder, both. So each of these vertical lines is an emission. The jaggedy line in the middle is just background noise. So chirp, 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 chirp.
Got it, aye.
You'll never forget it, huh?
You will never forget it now, will you?
So we're gonna do on the test, okay?
So the combination of the loudness and the close together of it, the time frame between you get the echoes back is really narrow, so that as that moth is moving, you have a very fine scale measure of the movements. It doesn't have a lot of time to move between each individual vocalization of the bat. Yeah.
So you know on this slide,
Yes.
how you showed that if you stimulate it loud enough,
Yes.
that it'll start to like not be able to sense it?
Yes.
Is that how they basically incapacitate the moths to be able to avoid them?
They don't. And that's the thing. They don't incapacitate the moths. So this is what a bat is doing. This is what a bat is doing, and this is what a moth is doing. A 1 cell and a 2 cells, and it's hard to see on this figure, but the figure in your textbook and on your screens is actually going to be better. But this is a B cell is.
Wait, was this in our textbook?
Oh no, this isn't in your textbook, but it's on there.
Okay.
You're going to see this much better on there than you will on this. So just pay attention to A1, which is the red dots. When this thing chirps, there's a response in the A1 cells that comes right after the chirp of the bat, and the lag between when the bat chirps and when the A1 receptor responds is determined by how far away the bat is. When the bat gets closer, it starts chirping more. And this is the end of this terminal buzz here, the end of the buzz. And the bat is very close. Chirp, activation, chirp, activation, chirp, activation, chirp, activation. You see that 1, 2, 3. This chirp has resulted in 2 depolarizations. This chirp has only resulted in 1. This chirp has actually come close enough to this depolarization that you miss a depolarization in here. So it is approaching the point where it's starting to get overstimulated, but it still remains active throughout all of this. What doesn't remain active throughout all of this is the A2 receptor. At these quick pulse responses and really intense levels, the A2 receptor basically just shuts down.
Wait, I thought you said the A2 only responded to them.
It does, but it doesn't keep it up. It's responding early on in the buzz, but as the buzz continues on, it basically stops responding at all. What this presumably triggers, and we know this from studying the moth, is that during this buzz, the activation of the A1 cell, rapid activation of the A1 cell, basically sends a message to the brain, and when that's processed by the brain of the moth, the nerve impulses, the motor nerve impulses that are sent out to the wings get disrupted. So normally, when a moth is flying, you have the brain, you have motor nerves going out to the thoracic muscles of the wing. These muscles operate the wing, and they're sent out in synchrony with one another. Wings up, wings down, wings up, wings down, wings up, wings down. When you stimulate A1, the brain interprets those things in such a way that when it starts to send out motor neuron impulses, it sends them out asynchronously such that it sends out contraction here, relax here, and then it sends out contract here and relax here. What this does for the moth, this will also help you remember, is during the time period when the bat is right upon the moth, the A1 receptor is signaling to the brain, and the brain is signaling so rapidly to the brain that the brain interprets that as, I need to switch from synchronous flight to asynchronous flight. And when you're flying like this, you're flying along in a straight line. As the bat gets closer, you're flying in a straight line faster, and as the bat gets right upon you, the brain switches because of the impulses from A1, and it goes from flat, fast, must get away, to whack, 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 whack. And when it does that, it starts this very unpredictable dive to the ground. So it goes from flying in a straight line to plummeting to the ground, but because the wings are flapping asynchronously, it's not very predictable. It goes, it's kind of like taking an oddly shaped piece of paper, except that it's too heavy. I need a lighter piece of paper. Jack, I'm taking a section of your paper. Well, actually, maybe a paper towel would be even better.
This is problematic.
It goes back and forth because it's not smooth. Wad it up so that it's symmetrical. Drops like a rock, drops straight down, very predictable. If you were ever in a place where bats were flying, so I used to go to Big Bend National Park, and there's this road that's right next to this campground that I used to stay at. And because it's lined with trees, the trees tend to funnel insects down this roadway because it's clear, and also tends to funnel bats down this roadway. And when I would go on night hikes, I could see the bats coming into this clear area, just going down the clear area. I would always carry a handful of pebbles, and as the bats got close, I would toss a pebble into the air, and you would see the bat, and he'd be flat, 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 flat. And then I would toss the stone up in the air, and then you would see the bat do this, go, and then he would figure out that it wasn't dropping like a moth drops, and he would go back to regular normal flight, because it was dropping like a stone, rather than dropping like a moth does. So you could fool a bat for a couple of seconds, but they figured out, that's not really a moth, that's not behaving in the right way, and then they would go back to their normal flight path. And I would just walk down the road tossing pebbles up at bats, watching them change their flight path, and then go back to normal flight when they figured out that it wasn't really a moth.
If you toss a little piece of paper towel, would moths try to attack it? You tossed it up and it was really unpredictable.
Oh yeah, they would probably follow it all the way down to the ground, or very close to the ground. So you can tether an insect with a really, really thin monofilament line and take quick pictures of it. Moths aren't the only things that can detect bat noises and respond to them. This is a lacewing, and what you see is this is the flight path of the lacewing, this is the flight path of the bat, and what you see is that when the lacewing gets receives the correct intensity of sound from the bat, it goes into this dive to the ground, and the bat attempts to intercept the lacewing as it's doing this dive. This is a series of high-speed photographs showing this.
Will you trace the bat's path one more time? Sorry.
The bat starts over here. Here it is, it's flapping along, and it's coming at it this way. Here it is coming up to try and meet the lacewing. It's pulling its wings forward to provide a larger capture area. And if it catches it, it's gonna envelop it with its wings, pull it into the body, and then catch it and eat it. But in this case, it actually misses it. Here's the lacewing after the bat has passed. It has basically, through this dive, been able to escape the clutches of the bat. So bat's here. No, it's a neuropteran, different order of insects. Bat, 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 bat. You see that the bat is coming back around, once again, to try and capture it on the flip side. So this is basically what moths are trying to avoid, is being captured by the bat. And lacewings also do this. They enter this asynchronous dive down to the ground. Once you're on the ground, you're lost in ground clutter, and the bat can't locate you. This is just showing sensory neurons go off to the brain, motor neurons go back out to the muscles, and it's the thoracic muscles that are basically located in the same area where the tympanum is. These thoracic muscles are the ones that go into asynchronous beating, asynchronous contraction, which leads to asynchronous beating of the wings, which then causes this chaotic dive to the ground. Now, why is this a predator-prey arms race? Because the predator is capable of exerting selection pressures on the prey. And in this case, the prey have evolved a set of behavioral responses that thwart the predator. And so presumably, as they get better and better at avoiding bat predation due to this behavior, that they then exert selection back on the predator to make the predator, the bat in this case, better and better able to locate flying prey. And we know that bats have amazingly good powers of echolocation. But in this particular predator-prey arms race, the moth may actually be winning. Because what they found in one species of moth is under the wing there is a structure on the exoskeleton, and the structure on the exoskeleton looks like a little file. Looks like a little file with all these ridges, and as the wings flap, these ridges can be engaged with a little spur on the wing, and as it does this, it creates ultrasonic vibrations that get emitted by the moth. So that when a bat is out there flying, when he's sending out chirps, he gets back a very clear sonic signal. The sound goes out, it bounces off things, and it comes back to him, and it's very clear. My sound hits something, and it's coming back in these very discrete sound waves. When a moth with a jamming file on its body detects a bat, it will engage the sound production of this structure, and what it does is it emits a series of ultrasonic chirps, such that the picture that the bat receives isn't this. It gets its own sound wave cluttered up with a bunch of moth sound waves. And when it does this, it makes it much more difficult for the bat to locate the moth, makes it more difficult for the bat to then catch the moth. So this is done, once again, from a set of experiments where they looked at a bat foraging on these guys, and the control moths are moths that are fully intact, and what you should predict is over a series of trials, night after night, the control moths should always get eaten at a relatively high rate because moths are good at doing what they do. If the purpose of this this apparatus is to just basically warn, like be something like an aposematic signal, early on in the first couple of nights, the bats should avoid it, but as they learn that there's nothing distasteful or dangerous about these things, they will learn that, and they will,
They would learn it.
Sorry, if it's aposematic, if it's warning, they wouldn't learn it, they would learn it, and their attempts to capture them would go down. If it's a startle response, they would be startled at the beginning, they would learn that it's a startling noise, and they would get better at capturing them night to night. And if it's jamming, actually interfering with their ability to echolocate, they would basically it would be harder uniformly for them to capture prey. And what you find when you look at the actual data is that the actual data for these are the controls. Controls got eaten a lot. These are ones that have the sound producing mechanism intact. And when you have your sound producing mechanism intact, you don't get attacked at all, or your capture rate is very low. And when you are a control, you don't you get captured a lot. So producing the sound appears to just uniformly make it more difficult for bats to locate you, so you get contacted less often, which means that you get captured more often. For sound producing moths that they left the sound producing mechanism intact versus in moths that they removed it from, basically smoothed that file out, when it was removed, you found that they once again approached almost 100% capture. And so what happens is as a bat is approaching, it increases the total sound output, but also the moth starts putting out a lot of sound. And what happens in this case is that these are the moth clicks, these are the bat clicks, and the dotted line shows an attack on a control moth for reference. And so as the bat is coming, it's clicking away. As the moth hears it, it starts clicking away. And in the case of the control moths that don't have a sound-inducing mechanism, the, wait a minute. Surplus and- Oh, this is what happens on a control moth. So, oh, on a control moth, yeah, okay. So this is a control moth. So this is pulse interval. The closer together the dots are, the more frequently the bat is putting out pulses. So there are a lot of pulses, and they start much earlier when you have a control moth. As the pulse interval goes down, you're putting out more pulses per unit time until you get to a point where you're putting out the maximum rate of pulsing that a bat can achieve, which is down here. When you have a bat that is chasing after a moth that is putting out a jamming signal, it takes them longer to realize that there's a moth there, so that their pulse interval doesn't go down until they're actually very close to the moth. Once they've either caught the moth or failed to catch the moth, the pulse interval goes back up. And so as pulse interval goes down, that means that they're clicking more, they're chirping more, and that happens earlier on in the interaction for control moths than for moths that have jamming apparatus. Alexis.
So what is the bat doing to evolve to become a better catcher?
And that's one of the things they haven't studied yet. This is relatively new stuff. When I was an undergraduate student, we knew about the reaction of the moth to the bat, but this is all stuff that's been discovered in the last couple of iterations of me teaching the class. And we don't know what if the bat is evolving in any way to counter this. Yeah.
So when the pulse interval decreases for the moth, the control moth, that means that the pulses, what you're talking about, those are the sonar, the ultrasonic?
These are the bat pulses. So what it's basically saying is out here, the bat hasn't necessarily detected the moth yet. You see how silent the bat is? Hasn't detected the moth. It starts increasing its pulse rate when it has detected the moth. And so as you have more pulses, more chirps per second from the bat, the pulse interval in milliseconds goes down.
So this is all the bat, this isn't the moths?
This is the bat on a control moth.
Okay.
This is the bat on a jamming moth, and the triangles are the jamming signal of the moth when it's a jamming moth. So the moth starts jamming the bat signal early. As it jams the bat signal, the bat doesn't respond the way it would if that jamming signal were not there. It still relatively slow chirps until it gets really close to the moth, and then it matches the control, but not until 150 seconds later in this process. And if 150 seconds later is enough to put off your interaction with the bat, that's possibly enough to get you out of the interaction altogether. All right. We will start on competition on Monday. We'll only get a little ways into competition. Do you want to still have the exam on Wednesday, or would you rather have the exam on Friday and get further into competition?
Wednesday for me.
Wednesday.
Okay, Wednesday it is then. Oh, here are your papers. Jack. Caleb. Caleb. Claire. Luke. Jalen is not here. Logan. Kennedy. Alexis. Are you wanting to talk to me now? Did you say something about
Yeah,
talking?
if you have time, it's better.
I unfortunately have a meeting. I'll be back at 2. So if you want to talk to me at 2, I can talk to you then.
It's like 3 of them, okay. What is the end when you go to the test? So 3 or something like that?
Sure.
You got mine.
Yeah.
Okay.